Hi, are you Dr. Johnson?
Yes.
Hi, Rhonda Jacobs. I'm going to be your interpreter for your class today.
Nice to meet you.
You too. Could you tell me a little bit what your class is about, what you'll be talking about today?
We're going to be talking about gene regulation, which is expressing certain genes, and we'll be talking about the lac operon in E. coli.
Okay, and briefly, what is a lac operon?
When organisms are living, they need certain gene products at sometimes, but not others. And in E. coli, they need to be able to use lactose sometimes, under some conditions and not others. The lac operon makes proteins that are used to use the sugar lactose.
Okay, and I noticed on your handout that you'll be talking about transcription. In brief, what is transcription?
Transcription means taking DNA, which is the genetic material, and you want to, say, make a protein from it. So what you do is you take DNA and you copy it over into RNA. That process is transcription. And then you take the RNA transcript and use that to translate to make the protein.
Okay, very good. All right, well, thank you very much.
Surely.
Okay.
We've been talking about DNA replication and RNA transcription. And what's important to remember here is that organisms don't transcribe all of their genes all of the time. What I mean by this is that there are some genes in the genome which are there, but for which you only have products some of the time. So if I could have the first slide. If you look at this slide, it shows you the elements that are in the genome that allow you to have a gene expressed some of the time, but not all of the time. For example, we have here that blue coding sequence that's going to code for a particular protein product. But we see next to it two kinds of regulatory or controlling elements. A promoter, which, as you remember, is where RNA polymerase binds, and a controlling site, a place where something, usually a protein, can bind and can regulate whether or not you have transcription taking place. So if we could have the slide off for a minute. So I'm going to talk to you today about a protein system in E. coli. It was the first well-studied gene regulation system, and it's called the lac operon. And what I'm going to do is draw a cartoon on the board of what's in the lac operon. So what I'm going to do is put down a whole series of letters, each of which has a specific meaning. P always stands for promoter. Now, there are different promoters for different genes, but P is a general term. So this is a promoter. In this case, the gene that's regulated right here is called the I gene, and it codes for something called a repressor. Then we have another promoter, and then we move down here and we have 3 genes, Z, Y, and A, that code for proteins. Z codes beta galactosidase. Y codes for something called permease. And A codes for something called acetylase. It's not as important. I'm not writing the name down. This O right here, what's that? Well, this is a controlling site. It is the operator. And what's this all about? Well, in E. coli, what we have happening is that E. coli usually grow on food where glucose is the main sugar. And the enzymes they need to break down glucose are always present. They're examples of genes that are always expressed. But you can put E. coli on media with different kinds of sugars in it, and it can use those different sugars. One of those different sugars is lactose. And the lac operon codes for enzymes that allows the cell to bring in lactose into the cell and to start breaking it down so it can use that lactose as an energy source. So going back to the board here, what we see is this permease gene, Y, is the enzyme that codes for the enzyme that actually helps to actively bring lactose in the cell. And the Z gene, beta-galactosidase gene, codes for an enzyme that takes lactose, which is a disaccharide, and breaks it into 2 separate sugars, so those sugars can be broken down. Now in the cell, what you find is if you're growing E. coli in a medium where glucose is in the medium, they will make hardly any of the Z, Y, and A gene products. But if you put them in a medium where lactose is the only carbon source, they will make large amounts of the Z, Y, and A gene products. What's going on here? How do they regulate it? And how does it all work? Well, we have a regulatory protein, and we have 2 controlling sites that we're working with. So what the cell is going to do is to use this regulatory protein. And if I could have the next slide, please. What typically happens is this I gene, the repressor gene, makes a protein. This is what's called a constitutive gene. And constitutive means that it's always around, it's always on. So this particular promoter allows RNA polymerase to make, as it turns out, small amounts of I gene RNA, messenger RNA, and that's translated into repressor protein. And so in this slide, those small pieces at the bottom represent the repressor protein. Now this repressor protein doesn't just want to float around in the cell, it wants to bind to something. And if you're growing the cells in medium with glucose, what it's going to bind to, what it looks around and finds to bind to, is actually the operator for the lac gene. And so the operator is the O site, and the repressor protein really floats through the cell and binds to the DNA in the O site. Now you'll notice that right next to the O site, that yellow site, is a green site, the second promoter. And promoters are for RNA polymerase binding. So what's happening there? Well, in fact, the RNA polymerase is binding. And it would, under some circumstances, be able to transcribe. But remember, we have repressor protein sitting in the yellow site there. So when the RNA polymerase tries to move down the chromosome, the repressor protein is sitting there like a roadblock, and transcription is blocked. And what that means is you aren't making any mRNA for these genes, and you aren't getting protein products. Now, I'm going to say the gene's turned off, a word of caution about that. In reality, when we talk about the repressor protein binding to the O site, what's really happening here is something not like us nailing a hook up on a wall to hang a picture. It's more like putting a Post-it note on it. Because what's going to happen is that the repressor protein will bind to the O site and it will sit there almost all the time. But once in a while it may fall off. And if there's a molecule of RNA polymerase sitting on the promoter right there, it will then transcribe this set of genes, Z, Y, and A. And it then will allow a product to be made. So you can detect a tiny, tiny amount of transcript and protein in a cell, even when it's grown on glucose. But I'm calling that off. Off isn't absolutely off. Off is practically off. The other thing to notice about that slide that was up there is that, in fact, there's only one promoter over there, and there are 3 genes. And you may wonder how in the world that's going to work. So let's think ahead here. If we go to the next slide, we're going to look at the situation that happens if you take your E. coli cells and you transfer them to a medium in which lactose is the only sugar. Now, lactose is the only sugar. What does that change? Well, there's a little bit, remember, a little bit of beta-galactosidase in the cell. There's a very little bit of permease in the cell. So if lactose is the only sugar in the medium, some of it can be brought in using the permease. And that lactose can actually be converted into a substance called allolactose. And allolactose is also what's in a very general sense called an inducer. Okay, so it's going to turn on expression of the lac operon. Well, how does that work? How do you turn on the expression of the lac operon? What you do here is you have the allolactose become involved with the repressor protein. So if you remember, I said the repressor protein wants to bind to something. It doesn't just want to be loose in the cell. It wants to bind to something. So if we look at the next slide, what we're going to see is that when allolactose is there, and that's that purple brown molecule, the little brown repressor proteins now have 2 things in the cell they could bind to. They can bind to that yellow operator site, but they will preferentially bind to the allolactose. And for that repressor protein, it's an either-or decision. That is, the repressor is going to bind to allolactose, or the repressor is going to bind to the operator, but it can't bind to both of them at the same time. So it will preferentially bind the allolactose. Now, if we go back to the slide for a minute, what you see is happening here is the repressor protein's bound to the allolactose. That yellow site is open and unoccupied. So when RNA polymerase sits on the promoter closer to the Z gene, it has nothing sitting in its way anymore, no more roadblocks. Transcription will occur. And when transcription occurs, you're going to make a messenger RNA. And the messenger RNA that the slide showed was, in fact, a messenger RNA that extended from the Z gene through the Y gene through the A gene. So in bacteria, we have something that I'm going to write on the board so you remember the term. We have polycistronic messenger RNAs. Okay? We really hadn't stressed that before. So what we're saying is that instead of making 3 messenger RNAs, we're making 1 messenger RNA that codes for 3 different proteins. And so we only need that 1 promoter and that 1 operator set to regulate the expression for all 3 genes. And that brings me to another thing. I called this the lac operon. So why is it an operon? Well, in bacteria, typically we see this kind of situation. And an operon, if I point to the board here, an operon is a set of genes that are put into this polycistronic mRNA plus their controlling elements that are right next to them. So the LAC operon is the promoter for the LAC gene, the operator, this is what I'm calling the laczine, Z for beta-galactosidase, Y and A. So in bacteria we have operons, and it's very, very common to just go ahead and regulate multiple genes at one time by one system of regulation. The controlling elements for the LAC operon then that I've talked about are actually three. There's the promoter, this promoter, the promoter of the LAC operon. There's this operator, and then going back over here, there's the repressor protein. Okay? And we call this an inducible system. Now, why is it inducible? Well, E. coli usually live in food where glucose is the principal or only sugar. They're adapted to that. They always make the enzymes for that. If you switch them to medium where lactose is the only sugar, you will increase the level of transcription and translation tremendously. You will have 10,000 times more protein if these cells are growing in lactose as the only sugar. And so we can turn this on under certain conditions. That's why we call this an inducible operon. Along with this idea that it's inducible operon, and you can imagine that some operons could be then repressible, things that were normally on but could be turned off. But this is an inducible operon, and we have a way to describe the kind of gene regulation that's occurring here. And this kind of gene regulation is actually, we would say that the repressor protein is a negative regulator, or it's negative regulation. So you ask yourself, well, why is it negative regulation? And the answer is, we classify this regulation by looking at the protein, the controlling protein, which is the repressor here. When the repressor protein is interacting with the DNA, what's it doing? Well, it's binding to the operator. That's what it does when it is interacting with the DNA. And it's a roadblock. It's blocking transcription or preventing it. So it's having a negative effect on transcription. So it's a negative regulator. Okay? Now, if we go back to the board for a minute, this is a very nice model. It was based on a lot of work. And I want you to understand some of the approaches that people can take to understanding how could they tell there was an I gene? How could they tell there was an O site? How could they discover these regulatory elements? And so what they did is actually look at bacteria under different conditions and grow them in different medium. So I'm going to put some things on the board as a summary of that kind of experiment. What they would do is we would have a normal strains, I plus, P plus. Plus just means normal in this sense. And what I've just said to you, what I could summarize this as, is if you grow this in glucose, that the Z, Y, and A genes are off. And I'm going to put a minus up there for off. And if we grew this in lactose, that the Z, Y, and A genes are on. Okay? Well, this is a genetics course. So if I'm going to tell you how they found these controlling elements, you're probably not going to be surprised to find out that what I'm going to be telling you about is how they worked with mutants to discover these elements. And I'm going to also try to tell you how they can distinguish different kinds of mutants. One kind of mutant that they worked with had normal Z, Y, and A genes, but those genes were always turned on. They were always above the off level. So going to the board, we have a mutant. As I said, it's in a strain with normal Z, Y, and A genes, but it doesn't matter whether I grow it in glucose or lactose. What's going to happen is that you're always going to get more beta-galactosidase, permease, and acetylase than you'd expect on your background conditions. And people thought about this and had this model, and they had two possibilities for why that might be. One of the first kinds of mutants they discovered was what's called an operator constitutive mutant. So in the operator constitutive mutant, everything is normal except the operator, this little controlling element. And so we say here, well, what's wrong with the operator? And the answer is that the operator sequence has changed. So imagine we're growing the cells in glucose. The cells are constitutively producing repressor protein. The repressor protein's looking for something to bind to. And if we go over to the board and we say, well, what's the repressor protein going to want to bind to? If we're growing them in glucose, all that's available is the operator. Okay, there's no allolactose there. It wants to bind to the operator. But when it binds to the operator, that binding is based on it recognizing a certain DNA sequence. And if that DNA sequence is changed enough, the repressor will fail to bind to the operator. Which means that when RNA polymerase attaches to the promoter, there's no repressor on that operator. That RNA polymerase can just transcribe right through. And that's exactly what will happen here. So if we look at the board and we say, okay, in this line, what I've drawn is when you grow it in glucose, you are getting more than what you'd expect from the off state of the Z, Y, and A proteins. That is turned on, and it's turned on because the repressor protein fails to bind to the operator, leaving the operator open, and you can get transcription then. When RNA polymerase attaches to the promoter, it will just read through. Now, of course, if we switch it to lactose, it's going to keep reading through. The repressor protein will finally have something to bind to, but that something will be allolactose. It's not even going to try to bind to the operator. It couldn't bind there anyway. RNA polymerase will bind to the promoter, and you'll get transcription. So that's one kind of mutant they discovered. A second kind of mutant they discovered involved that repressor protein itself. So I'm going to write this on the board too. We have a mutant, and I'm going to call this I minus. And I forgot to tell you that the OC is for constitutive, making these genes always on. Okay? The minus with the I means that you fail to make a functioning protein here. So I minus means the gene's there, but the protein it codes for doesn't work. Okay? So then we go back to the board and we say, let's keep everything else normal. What will happen? And the answer is, you know, this is going to look a lot like the mutant I just discussed, because we're going to make those gene products all the time. All the time. And if we stop and think, well, why is that? What's that repressor protein supposed to do? Well, if you're growing the cells in glucose, I always go through it in the same order. You start with the repressor, you say, do I make normal repressor? Here, the answer is no. I have non-functioning repressor. What do I mean by non-functioning repressor? It fails to bind to the operator. So if we're growing it in glucose, we can make repressor protein, but it, once again, is leaving the operator alone. The operator is sitting there open and unoccupied. When RNA polymerase attaches to the promoter, it will simply read through, and we'll get a transcript that will produce the Z, Y, and A gene transcript, and we'll get beta-galactosidase, permease, and acetylase made in glucose. Once again, if we switch it to lactose, of course, that defective repressor wouldn't have wanted to bind to the operator anyway. Once again, the operator is open, and you will have RNA polymerase attached to the promoter and transcribe the Z, Y, and A genes. So, the question from my point of view is, how can you tell it apart? How do you know whether you have a mutation in the repressor protein, an I minus mutation? how do you know whether you have a mutation in the operator, an OC mutation? And for that, we get into a nifty little genetic technique where we take our E. coli cells, which are haploid. They have one copy of their chromosome, and we make them partial diploids. We take the cell and we put another extra chromosomal piece in there that contains these genes so that we have a diploid. So if I put this on the board, what I'd have, and I'll put it on the top here, I'm going to have an E. coli cell with its own complete chromosome, and that chromosome includes all these genes I've drawn here. And then I'm going to add in the F factor that we talked about when we talked about conjugation. And this is what's called an F prime. If you remember, an F prime factor has a piece of bacterial chromosome on it. And so what we're going to do is we're going to have the bacterial chromosome, and then we're going to have an extra copy of a piece of that chromosome. And that piece is going to also have lac genes on it. So we're going to see the specific characteristics of that operator constitutive mutant in a diploid. So if I can have the next slide for a minute. This is a very busy slide, and I'm going to talk about it, and then I'm going to put something on the board afterwards. But first I'll talk. What this shows you at the top is that you have two chromosomes, and we don't really care which one's bacterial and which one's the F prime. What we care is that one of those two chromosomes, the top one, has a normal operator, normal repressor, normal promoter, but it is Z minus, which means that when you transcribe the Z gene, the protein you make doesn't do anything. It's non-functional. And then it has a normal Y gene and a normal A gene. The second chromosome, normal repressor protein, normal promoter, it has the operator constitutive mutant on it, then a normal Z gene, but now a Y minus gene. And once again, that means that when you transcribe that and translate it, the permease, the Y gene product you make, doesn't do anything. And then you have the normal acetylase gene. Now if I go to the board to show you what's going to happen when we go to glucose with this, it's really kind of neat, because I'm going to draw that second chromosome on top. We've got OC with Z minus, I think, and we have everything else normal. And then here we have O plus with Z plus and Y minus. Now, if we grow this in glucose and we think about what's going to happen, what we're going to get is the I genes. Both of these I genes are making perfectly normal repressor protein. And think about this. These repressor proteins are literally floating around in this cell, and they want to bind to something if they can. And so the repressor proteins are normal, and this is glucose that we're growing the cells in. So those repressor proteins are going to look for operators. And when they look for operators, they're going to find that the operator that I've drawn on the top chromosome is normal, and they can bind there. A repressor coded for by either chromosome can bind there. But the operator site on the bottom chromosome, that OC site, will fail to bind repressor. So we're growing these in glucose. And if we look at the board and we look at the top chromosome, what's going to happen is RNA polymerase will bind here, but repressor protein will be bound here. And so we will not transcribe the top chromosome. That's blocked. We go to the second chromosome, because there's two chromosomes in the cell now. And in the second chromosome, we have an OC mutation. And what we have happening is that RNA polymerase will bind to this normal promoter. Nothing, no repressor protein can bind here. So this is open, and you in fact will get transcription. And when you transcribe this, You'll make the whole transcript for the Z, Y, and A gene. But when you assay for function, do I have functional beta-galactosidase? Do I have functional permease, which are the two proteins you usually assay for? What you're going to find, if the situation is as I've drawn it on the board, is that when you transcribe the chromosome with Z minus and Y plus, you make a transcript for beta-galactosidase, but the protein product is non-functional. You make a transcript for permease, and the protein product is functional. So under these conditions, what you're going to see is that you'll be producing functional permease, but no functional beta-galactosidase. And what you can figure out from that is that only one of these two chromosomes is being transcribed. If both chromosomes had been transcribed, then between the two of them, you would make normal beta-galactosidase from the top chromosome and normal permease from the bottom chromosome. When you only find normal permease, that means only the bottom chromosome was transcribed. So we use this kind of partial diploid here to let us know that there are, in fact, different regulations going on in the two chromosomes. And we can say that the operators, then, are sometimes called cis-dominant. They control the expression of genes on the chromosome that they reside on. So if we look at the board again, when you have an OC mutation, you will get transcription of that chromosome even when you grow it in glucose. Because with an OC mutation, no repressor protein can bind here, and you will get transcription at all times. The operator is affecting the chromosome it's sitting on. On the top chromosome, we have O+. When we have this strain with both of these in the same cell and we grow them on glucose, normal repressor protein will bind to that operator, and it will block transcription on this chromosome. So each O is controlling transcription on its own chromosome. It's cis-dominant. Now if we switch this, if we change it so that we take this same strain, but we put it on medium with lactose, we have to think about, well, what will change? Well, what will change if we put it on medium with lactose is that now the lactose will enter the cell, a little of it is converted to allolactose, and that repressor protein will bind the allolactose. When that repressor protein is tied up with the allolactose, both operator sites will be open, and we will get transcription from both the top chromosome and the bottom chromosome. So when you look at the board, we're getting transcription now, both places, because both operators are open, both of them are open, and we will make Z plus product from up here, as well as Y minus and A plus, and we'll be making Z minus product, but Y plus and A plus down here. So when both chromosomes are transcribed, we'll get functional beta-galactosidase and functional permease. What about the situation where we have, in fact, an I minus mutation? Remember I said in a haploid cell, I minus and OC looked the same. But in a diploid cell, they can look different. So let's go back to the board again. If we look at the board, what I'm going to do, I'm going to leave this up in almost the same fashion. But I'm going to make one chromosome have I minus on it, and I will make both chromosomes be O plus. Okay. So now what we're looking at is individuals that have two copies of the lac controlling region and the lac operon, and they have a normal copy of the repressor protein, and they have a copy of the repressor protein that fails to make functional repressor. And we say, okay, let's plop these cells into medium with glucose. What's going to happen? Okay. Well, first thing we think about is that repressor protein. We always start there. One chromosome makes non-functional repressor, and that non-functional repressor binds nothing. It's as if it's not there. But the second chromosome makes functional repressor, and we're growing these cells in glucose. So that functional repressor is going to look for something to bind to, and what it's going to bind to is operator. Now we have multiple molecules of repressor protein. And so what they're going to do is those molecules are going to keep looking for some place to bind. Some of them will bind to the operator on the top chromosome, and some will bind to the operator on the bottom chromosome. So looking at the board, if we think about that, if both operators have repressor sitting there, then when RNA polymerase attaches to these promoters, well, we can't go through here, there's a roadblock. We can't go through here, there's a roadblock. So we will fail to get any transcription at all. And we will not see any beta-galactosidase, and we will not see any permease. Now that's different from what we saw with an OC mutant. And so what we've got here actually is something we're used to, something Mendel was used to. That is, we just have plain old dominance. What I'm telling you is if we have I plus and I minus in the same cell, that I plus acts like a dominant allele. And so the normal repressor protein made of one chromosome can control the expression from both chromosomes. It can block it. And so we have dominance, or sometimes people call this transdominance, controlling both chromosomes. Now if we look at the board real quickly, what we will see is that if we switch this to lactose, yes, we've got these two kinds of repressors. This repressor protein will still be made. But if you remember, in lactose, that repressor protein will find some allolactose in the cell. And that repressor protein, or those molecules of repressor protein, will satisfy their desire to bind by binding to the allolactose. So both sets of operators will remain open. And looking at the board, what we will see is that both operators will be open. RNA polymerase will be attached to both promoters, will transcribe both chromosomes, and we will make a transcript that produces functional beta-galactosidase and a transcript that produces functional permease. Both chromosomes will be expressed. So what I have shown you here is that we have controlling elements, that if we look at them in diploids, we can distinguish between different kinds of controlling elements. Things that make proteins are, in fact, show dominance. Things that are controlling sites show cis dominance. And this is true for all sorts of operons. This is true for inducible and repressible operons.